We were discussing about the gene interactions. So in the previous class, we talked about allelic or the non-epistatic interactions, which include incomplete dominance and codominance. In today's session, we will discuss about non-allelic gene interactions, which includes epistasis, that is dominant epistasis, and recessive epistasis. The second is the duplicate gene interaction. So where we will talk about the dominant duplicate gene interaction and recessive duplicate gene interaction. And the third is complementary gene interactions influencing the production of a final triad. And finally the dominant and the recessive gene interaction influencing the effect of the triad in the expression of the triad. Epistatic gene interactions. So what is this epistatic gene interaction? So gene interactions, what is gene interactions? Firstly we will see. So interactions between genes. So how many genes? Two or more different genes when it influences the outcome of a single triad. So here we are seeing homologous chromosomes, two genes which are present at two different loci. For example, we have taken gene B and gene B and gene G. So there are two genes present at two different loci. So the products of these two genes interact to produce the final triad. So the interaction between alleles of these two genes, interaction of the alleles of the two genes influences the expression of the triad. So that is called as the gene interaction, where two or more genes are involved in the outcome of a triad. So when two or more genes means, so each gene has two alleles. So the allelic interaction. So here these two, these are alleles of a gene, G, and this is alleles of gene B. But these are non-alleles. This is one gene, allele of one gene. This is the allele of second gene. So they are non-allelic gene interactions. What is epistatic? When the one gene masks or suppresses, masks or interacts with the product of gene B in expressing the final triad. So one gene masks the effect of the other gene. For example, here when gene G masks the effect of gene B, then it is called as epistatic gene interaction. So epistatic means the situation in which a gene masks the effect of the other gene. So epistatic interactions usually arises because the two genes encode the protein that participate in the sequence in a biochemical reaction. For example, if a colorless precursor is there, so two genes for the final purple color pigmentation, two genes are involved. So gene C codes for the enzyme C and gene P codes for the enzyme P. So if gene C codes for C, then this enzyme converts the precursor into an intermediate product. Further, this intermediate product is being converted into the purple pigment by the enzyme P. So if either of the loci is having a mutation, null mutation, none of that enzyme will be made. And so if, for example, here if C is having a mutation, then enzyme C will not be produced. So this colorless precursor cannot be converted into an intermediate. So therefore, this intermediate cannot be converted even if P enzyme is being produced to a purple color pigment. So here the pathway is blocked. Even if the second enzyme is mutated, if the second gene is mutated also, here the pathway is blocked. So if homozygous, if any of this path, any of the gene is mutated, then the product, there will be a altered product, or no product will be formed. You have other term called as complementary gene action. So what is this complementary gene? When two genes work together to produce the final product. For example, here we will take gene C and gene P. So gene C produces an enzyme C, so that converts the precursor into an intermediate product. And the gene P in turn will produce enzyme P, which helps in converting the intermediate product into the final product, the colored product. So here both the genes work together to produce the final product. This type of action where each gene complements, they work along each other, they complement each other. This is called as the complementary gene interaction. Duplicate gene action. The other type of gene interaction we see is duplicate gene interaction. So here you have a precursor, a starting product, which is converted, starting substrate, which is converted into a product. So here there is two genes, one gene is B and the other gene is A. So gene A produces an enzyme which will convert the precursor to product. Same way, same pathway is followed by this gene B also, producing enzyme B, which converts the precursor into the product. So both the actions are duplicate actions, redundant actions. So that is called as, what is it? Duplicate gene action. They duplicate each other. When the two pair of genes have identical function, they are doing the same function, similar function, that is called as duplicate gene action. So these genes, that is the duplicate genes, they can be, let us take two genes, what it means is they are two identical genes A and B, which is going to produce the product with the identical function, following the same pathway. Okay. So here two identical genes producing the same phenotype. So these two genes can be present on different loci on the same chromosome, like what is seen here. Let us take chromosome 1. So it is, one is present in P arm and the other one in Q arm, P arm and Q arm, or you can take, or it can be present on a different chromosome. So A is present on chromosome 1, whereas B gene is present on chromosome 8. So these duplicate genes can be present on different loci on the same chromosome or present on the different chromosomes. Yes. So moving on to the gene interactions and what is the type of offsprings we get. So we will see in detail about each of the gene interaction. So first we will start with dominant epistasis. So we saw what is this epistasis. Epistasis is where one gene masks the effect of the other gene. Here this masking effect is because of the dominant allele of one gene. So that is why it is called as dominant epistasis, where one dominant allele of one gene will mask the effect of alleles at the second gene. So here what is the ratio we get is 12:3:1. So we will take first case, that is we'll take an example, pigmentation in fox glow flowers. So here the foxglove flowers, they get the pigmentation because of interaction of two genes, that is gene 1 D and gene 2 is this W. What is this gene D? The gene D is responsible for the color, that is the pigmentation. If one allele gives a dark red pigmentation, whereas the recessive small d allele produces a light red pigmentation. W gene, the second gene W, what is the role of this gene is the deposition of the pigment on the flowers. The dominant gene W, capital W, what it does is it restricts the pigmentation to a small area in the flower, that is in the throat of the flower. You can see here in the throat of the flower. So W confines the pigment to the throat of the flower, whereas the recessive W, small recessive allele, small W, it doesn't confine it, so the entire flower will be colored. So here when the presence of this dominant allele, dominant one dom, that you have two genes, out of which gene which confines the that is deposition of the pigment, which is controlled by this W. If the dominant allele will mask the effect of the other gene D or small d. Even it is producing the color, it will be only pigmented at a specific, it will be restricted to the specific area in the flower. Whether D will produce dark red, even if it is producing dark red or it is producing light red, when the dominant allele of the second gene is present, the pigmentation will be towards the throat of the flower. Only when it is in the recessive condition, the flowering, but when it is in a recessive allele is present, only the pigmentation will be throughout the flower. So here when capital D, dominant D allele is present, so it will form red color and the pigmentation will be throughout the flower. When small d, light red color pigmentation, so here you have the recessive allele of the second gene, so here the pigmentation will be throughout the flower. So this is an example for dominant epistasis, where dominant allele of one gene masks the effect of the other gene. So here, yes, so we have talked about the triad, the flower pigmentation. So here this triad is controlled by the interaction between two genes. One is the D and W. D gene controls the intensity of the color produced and W restricts the pigmentation. D gene has two alleles, dominant and the recessive. The dominant allele is responsible for a dark red color pigmentation and the recessive allele small d is responsible for light red color. W gene, it has the dominant dominant allele responsible for restricting the pigmentation to the throat of the flower, whereas the recessive allele does not restrict the color, so the entire flower will be colored. So we will so here in the dominant epistasis, dominant allele W gene masks the effect of the other gene, that is whether it is in the dominant or in the recessive form. Hence it is called, it is a good example of dominant epistasis. So we have done a cross. So the parent, it is homozygous, we take parent, the gametes what we get, and in the F1 we are crossing the heterozygous parents. So we draw a Punnett square, and what is the ratio? So ratio what we get, three types of phenotypes. That is, one is the white color flower with all the pigmentation or restricted to the throat in the flower. Then you have the dark red pigmented and the light red. So you have three phenotypes. So the dominant here is this throat pigmentation, so 12 is to 3 is to 1. The next one is the red is the third, and the red phenotype is 3, and the recessive phenotype, that is light red, is 1. So the phenotypic ratio or in dominant epistasis is 12 is to 3 is to 1. The next one is the recessive epistasis. So here epistasis, that is one gene is going to mask the effect of the other gene. But here what gene? That is, okay, one gene, you are having one gene. Let us take here B and E. But here, which, that is the recessive allele of one gene, it is going to mask the effect of the other gene. So we will see an example here in Labrador Retriever coat color. So if you see the triad here, that is the fur color, it is controlled by the gene interaction of two genes B and E. B gene has two, and that is has the two forms. One is the dominant form, which codes for the black pigmentation. The recessive allele B codes for the brown pigmentation. The second gene, E, controls the deposition in the hairs. So, dominant allele, capital E, allows the deposition of the pigment. But the recessive allele, small e, prevents the deposition, or there is no deposition of the pigment on the hair. Therefore, if you see the fur color, it will be yellow color. So here the allele, recessive allele of gene E, that is which controls the pigment deposition of the pigment, is epistatic to the gene, that is pigmentation gene, dominant or in the recessive form. So we are taking a cross. If you are crossing the heterozygous parents, then you draw the Punnett square, see what is the outcome. If you take here, you have the black coat. So the black coat is 9 and the light brown is 3 and out of 16, and yellow color is 4. So the phenotypic ratio will become 9 is to 3 is to 4. So why is this black color? Then that is, you have the B and the B allele, capital B, dominant B, and the dominant allele of both the genes are present. So here black color pigmentation will be produced and the deposition is also done. So the fur coat will be black. Brown color, the recessive allele for the pigmentation is present, so it will be producing the brown melanin pigment, and you have the dominant allele for the deposition. So deposition will take place. So the brown pigment is produced and the deposition will take place. So the final fur coat will be brown in color. And here, when the recessive allele for the deposition, that is small e, is present in a homozygous condition, it is going to mask the effect of the color producing pigmentation gene, whether it is in the dominant form or in the recessive form. So there won't be any deposition of the pigment. So here you can see, even if the pigmentation gene, if it is in the dominant form, if the second gene for deposition is in the recessive form, homozygous, then there is no deposition of the pigment. So the fur coat will be yellow. And here, if the pigmentation if the pigmentation gene is in the recessive allele form, also the deposition, that is, it can produce what is that brown color, but no deposition will take place. So here no coloration, so yellow color coat only we will get. So this is the recessive epistasis, where recessive allele of one gene masks or reduces the expression of the allele in the other gene. So the ratio here what we get is 9:3:4. Recessive epistasis is also known as supplementary gene interaction because supplementary gene interaction is seen in recessive epistasis. What is a supplementary gene? So here they are non-allelic genes, that is two genes. So out of the two genes, one gene, let us take first gene, one gene, gene A, it can produce the phenotype, that is the effect, even if the second gene is present or not. But the second gene cannot produce the effect in the absence of the first gene. So it can produce the effect only in the presence of the first gene. Therefore, it is known as the supplementary gene. Let us understand this concept by studying the gene interaction in mice coat color. Usually the animal fur coat is determined by at least by 5 genes, that is A, B, C, D and S. For our study, we will see the gene interaction between A and C, which is responsible for this hair color pigmentation, that is one agouti hair color and second one is this black pigmentation. Agouti pigmentation is the wild type which is found in most of the animal coat hair color, and black is the recessive type. When we see the individual hairs on the mouse coat, we see these two type of hairs, that is agouti hair and the black hair. The agouti hair, you can see it is having a black pigment, but a small yellow band is found. So yellow band on this black hair gives this brittle appearance. So overall, if you see the overall fur coat color of this agouti mouse will be grayish color. This gives this yellow band in the black hair. It gives this brittle appearance, that is the salt and the pepper appearance, which overall if you see it will be a grayish coat color. And the black pigmentation is, if you take the black hair, the entire hair will be having a solid black pigmentation. The yellow band will be absent in the black color. So gene A, the two genes which is controlling the that is this black pigmentation and the agouti hair color is controlled by two genes, gene A and C. So gene A is responsible for the distribution of the pigmentation. So the dominant allele A is responsible for the agouti phenotype, and the recessive allele small a is responsible for the solid dark black pigmentation throughout the hair. So this black hair is because of the recessive allele, and the agouti pigmentation is because of the dominant allele capital E. The gene C is responsible for the color production, that is color expression. So the dominant C permits the color expression, whereas the recessive C, that is the recessive allele, prevents the color expression. The metabolic pathway for the supplementary gene interaction seen in the recessive epistasis is, we have a substrate which is converted into a colored pigment in the presence of a dominant allele of the C gene. So this dominant allele of the C gene produces an enzyme which will convert the substrate into the colored product. But the recessive form does not does not produce the enzyme. So here the color, that is substrate, will not be converted into the color pigment. So once the that second gene A is responsible for the distribution. So here the dominant allele A is responsible for the agouti phenotype, and the recessive allele small a is responsible for the black phenotype. Therefore, gene A and C interact to produce the agouti phenotype. That is, here the gene C supplements gene A in producing the agouti phenotype. That is why we tell supplementary gene interaction. Therefore, the recessive epistasis exhibited by coat color genes in mice is, we call it as supplementary gene interaction. The same applies to the dog coat color also. So if you see here, the effect, final effect, what you get is dominant epistasis, that is, and here the gene interaction is one gene masking the other. Same way in recessive epistasis, here also it is a supplementary gene interaction where you find a recessive epistasis. That is why we call it as recessive epistasis or supplementary gene interactions. So one example we have taken is the mice coat color. So we can see what is the final phenotypic ratio is 9:3:4. The next type of gene interaction is duplicate dominant or duplicate gene action. So what is this duplicate dominant? So here we are using two terms, duplicate and dominant. So what is duplicate? When two genes, we have seen already what is this duplicate, when two gene pairs have identical functions. So they two genes, for example here we will take gene P and gene R. So P also P gene produces an enzyme which is converting the precursor to anthocyanin. The next gene R also produces an enzyme which converts the precursor to anthocyanin. So two genes which are doing the same function. So they have duplicate gene action. So they have a duplicate gene action. And what is the gene? That is the dominant allele of both the genes. Okay, dominant allele of both the genes have duplicate gene action. So here, genes in a redundant system have duplicate gene action. They code the same product. So if either of the dominant gene is present, allele of the gene is present, then they will be producing the same product. But if they are mutated, both the genes are mutated, then you will have a mutated product. So here duplicate. So two genes doing the same function, duplicate function. And here that is what we are talking about, the dominant allele of both the genes. So dominant allele of both the genes will, if either of the genes dominant allele is present, it is going to produce the product. So for example, you see here P, capital P, that is dominant allele of the P gene is present, then it produces the purple color. In the third condition, you can see the P gene is recessive, but the second gene R, it is of the dominant allele. Then also the product will be formed because they are having identical functions. But when both the genes are having the mutated allele, then no pigment will be formed. That is this duplicate dominant gene action. So duplicate gene, I have already told you, when two identical genes. So it is like they are two identical genes which are producing the same phenotype, and it is meaning that two genes which are present on the same chromosome but at different place, different loci, or two genes, identical genes which are present on two different chromosomes. The thing is they are identical, so their function will be similar. That is why we are calling it as a duplicate gene. The fourth one in type of interaction is duplicate recessive. So here these genes, what type of gene interaction they are going to have is duplicate gene interaction. And where do you find this duplicate reaction? In the recessive genes, in the recessive alleles of the two genes. And another one thing is we have complementary. It is also called as complementary gene interaction. So why it is called as complementary gene interaction? First we will see about these genes. So these are two genes. So for a triad is controlled by two genes. Okay, they are non-allelic. They are not alleles of the same gene. So they are two genes. So they are non-allelic genes. That is two genes which act together to produce a product. So you have, let us take here, there is a gene C and there is gene P. So they act together to produce a triad. But absence of either of this, if P is, so both the genes in the dominant form has to be present for a particular triad to be produced. If either of C or P is absent, then the product will not be formed. So that is why we are telling the gene action interaction is complementary. They work together. They compensate. Okay. They help each other and they produce a product. So that is why they are complementary gene interactions. But why we are calling duplicate recessive? So duplicate we know that is two genes doing a same function, identical function. So C is also producing a product and P also produces the same product. So both the genes are producing identical pheno, that is producing identical product or the phenotype. But here the thing is we are using recessive. So what is the gene? Whether the dominant allele is doing the duplicate function or the recessive allele is having this duplicate function. So if you see here, it is the recessive allele pair of either of the genes, okay, produce the mutant phenotype. So here either the mutant allele of the C gene or the mutant allele of the P genes, when they are in the homozygous condition, they mask the effect of the dominant P or the C and they produce a mutant phenotype. That is why we call it as a duplicate recessive. So we call it this type of gene interaction. It is complementary, that is two genes are involved. So two genes complement, work together to produce a product. But when either of the gene is absent, when either of the dominant allele is absent, of the genes is absent, then a mutant phenotype is produced. So here Otherwise, we can tell if the recessive allele of either of the gene is present, then a mutant phenotype is produced. Let's take in this case, so both the dominant allele is required for a product to be formed. So here in this case, sweet pea flower color, you have gene C and gene P. So gene C product, what it does, it converts the precursor to a precursor, intermediate, precursor 2. So further the precursor 2 is converted into the pigment by the product P. Gene P product is converting this precursor 2 into the pigment, anthocyanin. So both has to be present, that is dominant alleles of both the genes has to be present to produce the colored product. If either one dominant, that is either one dominant is allele of either of these genes are absent, then the product is not formed. Let us take this case where the dominant allele of C gene is present but absent in the case of the second gene P. So here no, that is it is converted into precursor 2, but further conversion will not take place, so no pigment is formed. The other case will take when the gene C is not, it is, that is the dominant allele is absent but the mutant allele is present, then also the precursor, no, first precursor one itself is not be converted, so no pigment will be formed. So here duplicate, whether it is in the dead or the other way, that is the recessive allele, whether recessive allele of one gene or the other gene, when it is present, it is going to produce the same phenotype. That is why we call it as duplicate recessive. So what is the ratio we get in the F2 generation is 9:7. So here we have taken it in the sweet pea flower. So a cross is carried out because between the heterozygous parents, and you draw you Punnett square and work it out, and what is the ratio you get is 9 purple colored is to 7 white colorless flowers. The next gene interaction is duplicate dominant. So here duplicate we know. So here the genes, so two genes are involved. So let us take here gene 1, that is two genes are involved. One gene is P and the second gene is R here. So two genes are involved, and here what is that? The dominant one. So here we are talking about the duplicate action of what allele? Dominant allele. So here dominant allele of the both the genes. Okay, so here they are having duplicate action. So here P, when it is present in the dominant form, it can produce the product. If the other gene, it also has the same pathway, so it will be producing the product. So if any one of the dominant gene allele is present, any one, if the dominant allele of either of the gene is present, then the product will be formed. So here in the second case you can take, so the dominant allele of P gene is present, so it can produce the product. The third one you can see P is in the recessive form, whereas R gene is in the dominant form. Then also it can form the product. Only when both the genes are in the recessive form, a mutated product is being formed. So that is why it is called as duplicate dominant. And the ratio what you get is 15:1. So here example for a cross we have taken, that is shepherd purse weed capsule shape. Okay, so here either it is triangular or oval. Triangular shape is controlled by two dominant genes T and D independently. So you perform a cross and you see here. So T and D are two genes. So here if either one is present, then the product will be formed. But if both are mutated only, then only you will have the mutated phenotype. That is the ovid. Otherwise, you will have triangular phenotype. So the ratio, final ratio, what you get is 15:1. The next gene interaction is duplicate genes with cumulative effect. So here what is the action is duplicate genes. So two genes are there. Okay. So let us take here two genes A and B. So both are going to produce the same product. So duplicate genes, that is two gene pairs with identical function. So here, so two genes which are having the identical function. But here both the genes, when they are working together, we are going to have an increased effect. That is what additive effect, additive gene interaction. That is why it is called as cumulative or additive effect. So here let us take, in this case, you have shape of this squash fruit is being determined by two genes A and B. So here we told duplicate the genes. So if A is also going to follow the same pathway and produce the protein, that is A is going to convert the precursor into protein A, which will turn will form a disc shaped fruit. B gene will also in its dominant form produce protein B. This protein is also going to produce the disc shaped. So both A also produces same disc shape. B also produces this final same phenotype, same trait. So either one, if A is present, also it's going to, but here, but the thing is, when both are present, then it is going to have a cumulative effect, increased effect, additive effect it is going to show. But if only one copy is present, okay, it is going to produce, but here the effect will be less. That is what it means. The molecular mechanism behind it, what we can come to know is, that is this product, final product is by 2 units, let us take. So here when both the genes are in the, both the dominant alleles are present, then both the units are produced and you have an additive effect. But when only one dominant allele of only one gene is present, then one unit is produced and the effect is less. So you have a different phenotype. But the thing, at the same way here, in the B also only one unit, so you have a, what is that, different phenotype, that is spear shape. But when both units are present, then both are added additive effect, so you get a disc shape. So that is why we call it as a duplicate gene. So duplicate action, but when both are present, you have a more effect. It is additive effect, cumulative effect you get. Okay. So when both are in the recessive form, then you get a different altered phenotype. So what is the ratio you get is 9:6:1. So duplicate genes with cumulative effect. So here also we have two non-allelic genes. So duplicate, so they are having similar effect. So both A also will produce same effect, B also same pathway it is going to, that is same action it has and the same phenotype it is going to have when they are separate, when they act separately. But when both the genes come together, they have an enhanced effect. That is why additive effect or cumulative effect we tell. Molecular pathway behind it, that is the dominant alleles of each gene produce one unit of the product. That is A and B, when both are present, 2 units are produced. Therefore, it leads to enhanced cumulative effect. But when both the genes are in the recessive form, no product will be produced. So, an altered phenotype or a muted phenotype will be expressed. So, here, what is this is a cross for the squash fruit shape. So, the final ratio what you get is 9:6:1. to 1. And finally, we see this inhibitory gene action. It is also called as dominant and recessive interactions, or we call it as a inhibitory gene interaction. We also use a term called as dominant suppression. Why do we use all these? We are telling it as dominant recessive epistasis. Why? If you take here, a triad is controlled by two genes, gene A and B. So here gene A in its dominant form is epistatic to the other gene B. So it shows a dominant epistasis. But when the other gene B is present in its homozygous recessive form, it is epistatic to A. So here in this gene interaction, you can see both the dominant and the recessive allele epistasis. That's why you're calling this gene interaction as dominant recessive epistasis. And why are we using the term inhibitory gene? Because here we see inhibitory action of a dominant allele of one gene over the expression of the other gene, even if it is present in the dominant form. For example, we will see in the chicken feather color. So if you take in the chicken feather color, there are two genes involved. One is this gene C and gene I. So in the gene C, the dominant allele capital C is responsible for the pigmentation of the feathers. But whereas the recessive allele does not code for the pigmentation and results in white feathers. The second gene I, the dominant allele capital I, can suppress the color producing effect of the dominant allele of the gene C. So here the gene I can suppress the color producing effect of the C allele, that is even in the dominant form. Even if it is present, it can be suppressed by the dominant allele of the other gene I. So it is showing an inhibitory action. That is why we are telling inhibitory gene action. And here the inhibitory action is by a dominant allele of one gene over the other gene. So that is why we are telling here it is dominant. We also call it as dominant suppression. So the ratio, what the phenotypic ratio what we get is 13:3. So we perform a cross and we put a Punnett square and find out what is the final phenotypic ratio. The final phenotypic ratio what we get in this dominant suppression, or what we call it as inhibitory gene action, or dominant recessive epistasis. The phenotypic ratio what we get is 13:3. So these are all the different gene interactions observed in different organisms.